ovum, today we will be learning about ovum. Ovum, it's produced by the female, whereas the male produces what is known as sperm. The ovum, it has the basic structure of cell. The basic structure of cell, it includes what all is there in a cell. It has a nucleus, a nucleolus, cell membrane is there, as well as cytoplasm. Here also you find all these organelles, and the cytoplasm of an ovum is known as ooplasm, whereas nucleus is known as germinal vesicle, and nucleolus, that is known as germinal spot. So there's a bit change in just the name. The rest of the structure, the organelles, all these are same. And ovum gives the nutritive factors to the growing embryo. Sperm gives the pronucleus, whereas the ovum provides the nutrition or the nutritive factors. So these nutritive factors should be present within the ooplasm, and this is present in the form of yolk. Yolk is present within the ovum, and the ovum is produced within the ovary. Ovum, that is produced within the ovary, and it transports from the ovary to the oviduct, and once fertilization takes place, it reaches the endometrium. So starting from the ovary, it is known as ovum, and it passes through the uterine tube and reaches the endometrium after fertilization. After fertilization only it reaches the endometrium. If fertilization doesn't happen also, it reaches the endometrium. And the zona pellucida and the spaces, this ovum is covered, in addition to the basic structures of cell, it is covered by few membranes. You could see the structure. This is the structure of ovum, which has a nucleus, a nucleolus, as well as a cytoplasm, and it has a cell membrane. A cell membrane is present, and that's a basic structure. After the cell membrane, you have a vitelline membrane. A vitelline membrane covers the cell membrane, and the region in between the cell membrane and the vitelline membrane is known as perivitelline space. It's known as perivitelline space. Here, the plasma membrane covering this ovum, that is projected into numerous microvilli. Outpushings of these plasma membrane are there, which is known as microvilli. Plus this region, it has a rich supply of cortical granules. These cortical granules are produced by Golgi complex. Golgi complex produces cortical granules, and this cortical granule, it plays a major role. Once sperm enters, there's a shift in this perivitelline space. The perivitelline space detaches from this envelope, the other membranes, and then the cortical granule. It provides us a thick covering preventing polyspermy. Polyspermy is entry of further sperm. And coming to the structure of ovum, you have the cell membrane, the perivitelline space, the vitelline membrane is there, and then you have what is known as zona pellucida. Zona pellucida is the next layer that is present. It's another type of ovum. Ovum, structurally it is different in different phylum, and here you could see that space clearly. The perivitelline space is there, and there you have the first polar body. Meiotic division takes place. The first meiotic division takes place when the ovum is within the ovary, but once it reaches the oviduct and fertilization takes place. If fertilization takes place, only second meiotic division happens. So when the sperm touches the perivitelline space, or the vitelline membrane, then there's a rearrangement, and this rearrangement brings about second meiotic division. Till that time, within the ooplasm, you have the nucleus, which is known as pronucleus, or that nucleus is within the has just completed its first meiotic stage, whereas the first meiotic stage produces two the oogenesis produces two oocytes, and one remains within the ooplasm. The one that remains within the ooplasm, that's a dominant one, which is destined to become the embryo, whereas the second one, that is smaller one, that is known as polar body, and this remains within the perivitelline space. And then you have the zona pellucida covering it, and then an outer covering is also present, which is known as corona radiata. Actually, this is what happens when the ovum is released at the time of ovulation from human embryo. When it is released at this time, you have such a structure which is known as ovum. It's not called an egg, it is known as an ovum at the time of ovulation. Once it enters into the oviduct, it is within the oviduct that fertilization takes place. Sperm that enters into the body, it climbs from the endometrium and tries to reach the oviduct. The sperm that reaches first and touches this vitelline membrane, that fertilizes, that moves within and it fertilizes, and the rest of the sperm that just gets degenerated. So it's just single sperm that fuses or that undergoes fertilization, and that results in what is known as zygote. Zygote is resulted. So these are protective covering, the zona pellucida as well as the corona radiata. These are protective coverings of the ovum. And egg envelopes, these egg envelopes, they could be divided into three: the primary egg envelopes, the secondary one, and the tertiary egg envelopes. Egg envelopes, they are also known as egg membranes, and the primary one, that's a zona pellucida. That's a zona pellucida, or in certain cases it is known as zona reticulata. Whereas the secondary one, so the first one, the zona pellucida, that is secreted by the ovum itself. The ovum itself secretes that layer. The second one, that is known as corona radiata or chorion in insects, and that is secreted by the ovary as a protective layer over this ovum. And the third one you usually find in forms where the the fertilization of the egg depends on mercy of nature, where it requires additional support. So you have a jelly coat in the case of frogs. A covering is there which is jelly like, and once the frog egg reaches water, touches water, what happens is the gel tries to take this water, it swells, and this jelly coating becomes a protective covering. And then you have albumin, shell membrane, and shell. These are tertiary egg membranes in the case of chick or birds, where they act as protective covering. Once shell membranes are added, or these egg membranes are added to the egg ovum is known as egg. It's at the time when these membranes are added, the ovum is known as egg. And significance of egg membrane, the first thing is egg membrane, it's within the egg membrane that sperm attaches, so it helps in sperm addition, prevents polyspermy. There's changes once the sperm enters, a single sperm enters into the ovum, there's changes within this vitelline membrane and the perivitelline space, And what happens as a result is polyspermy is prevented. And the cortical granules play a major role for the prevention of polyspermy. And then it's a protective covering, you know, the shell of hen. Actually, it's a protective covering from all environmental hazards. And again, a definite shape is maintained because of these egg membranes, which help in blastulation or proper cleavage. And once again, the ovum, coming back to ovum, the nutritive purpose of ovum, the nutrition or the growth of the zygote, that depends on the nutritive factor that is present within the ovum. And much of this maternal complements, they add to the growth of the ovum. And the cytoplasm of egg, it is known as ooplasm, where you have all these cellular organelles, and in addition to this you have yolk within the ooplasm, and this yolk could be distributed based on the phylum. The distribution pattern is specific for each phylum. And there are numerous microvilli which are outpushings of these cell membrane of the ovum, and they contain numerous mucopolysaccharides. And coming to polarity, there's polarity is something that you find in eggs. In egg you have an animal pole as well as vegetal pole based on the presence of yolk. Yolk is present, where yolk is present more, it is known as vegetal pole. And the opposite side where growth takes place, that is known as animal pole. Maximum metabolism that takes place within the animal pole, whereas the yolk, that's just a supplement of energy. Yolk, you have a reserve of energy, the energy being utilized for metabolic activities of the growing embryo. The embryo is found towards the animal pole, then you have a plane passing. If animal pole is on top and the vegetal pole is below, you have a plane connecting these two poles, and within this axis which is known as polar axis lies a nucleus. And mostly when the yolk is more, what happens is the nucleus is displaced towards the animal pole. You have an animal pole and a vegetal pole. The nucleus is more oriented towards the animal pole. And egg, it's classified based on the presence of yolk and development. And on the basis of amount of yolk might be less, it may be absent, it might be less, it might be concentrated towards one portion, and based on the amount of yolk you could divide it into 4. It's divided into 4 classes: alecithal, microlecithal, mesolecithal and macrolecithal eggs. Alecithal, microlecithal, mesolecithal and macrolecithal. From the word itself, alecithal, where yolk is completely absent, and this occurs in case of certain eutherian mammals. So the mother provides a nutritive factor, so there is no need for extra nutritive structures, that is why yolk is absent. Then comes the microlecithal one, like Amphioxus tunicates, where some amount of energy is required, and this energy is provided by yolk that is present scattered here and there. And example for microlecithal, or it's also known as oligolecithal, because the amount of yolk is less. Then the next classification is mesolecithal egg. Mesolecithal egg, here in amphibians, the amount is much larger and it is found scattered throughout the ooplasm. Then if the amount is larger than the microlecythal, it is known as mesolecythal. And then comes the macrolecythal or megalecythal. It's known as macro or megalecythal because yolk is present in large amount. And mostly terrestrial forms, they have this sort of egg where the amount of yolk for development, that is large. For completion of that embryo, you require all nutritive factors, and these nutritive factors are present within the yolk. So example, you could go for reptiles. So these are the 4 classification: alecythal, microlecythal, mesolecythal and megalecythal or macrolecythal, based on the amount of yolk. Then next classification, it's based on distribution, how it is distributed, whether they are evenly distributed, whether they are based on whether they are concentrated towards the vegetal pole. It could be divided into isolecythal or homolecythal, where yolk is scattered throughout evenly. And example for this is Amphioxus, mammals comes as an example. And the next classification is telolecythal. Telolecythal, a large amount of yolk that is present and that is concentrated towards the vegetal pole. And mostly everything comes in this telolecythal egg, where you could divide it into 3 types, and that is slightly telolecythal. Now it's not the tail actually. Actually here when we are saying telolecythal, it's just because it is more concentrated towards the vegetal pole. The vegetal pole is being considered as tail. That is why we are saying it as telolecythal. It's slightly telolecythal where it is eggs of fish, where the cytoplasm towards the animal pole is clear and you have a very little yolk which is placed towards the vegetal pole. And then you have moderately telolecythal one, which is characteristic of amphibia. Yolk plays a major role when it comes to cleavage. When cleavage occurs, the clear cytoplasm or the metabolic point that divides faster when compared to the vegetal pole. Vegetal pole, that division is bit slow. There mostly it's fat deposits that's present, so there the division is bit slow. And this cleavage pattern changes because of the distribution of yolk. And moderately telolecythal one, again it has yolk within the vegetal hemisphere, and then you have the vegetal hemisphere, you have the nucleus that is shifted towards the animal pole. And example is amphibian one, and extremely telolecythal one, where you have the reptilian ones where yolk content is more, mostly that animal pole, there you have a free area and the rest is just bulky yolk, and that forms the extremely telolecythal one. An exception to this is centrolecythal egg. Centrolecythal egg that is found within insects. Insects you find the centrolecythal egg, and the centrolecythal egg, it's like you've seen, it's a spherical, more or less ellipse in shape, and within the center you have the nucleus. Within the center portion you have a nucleus, and this nucleus is completely covered by a yolk, and you have a thin cytoplasm. At the peripheral region. So within the center you have a nucleus, a yolk surrounding it, and surrounding it you have a thin cytoplasm. Then that is known as centrolecythal egg. Here cleavage pattern is entirely different. It's entirely different from the rest, where this nucleus has to migrate outside towards the cytoplasm, because only nucleus is present in the center, the rest everything is occupied by yolk. And then you have a thin film of cytoplasm at the outer end. And then you have another type which is known as mosaic and a regulative egg. Most of the experimental studies in embryo is based on this mosaic and regulative eggs. Mosaic eggs are nothing but if during cleavage it cleaves into equal halves, a single cell zygote forms into a multi-level organism where the number of cell is more. From a single cell you get an organism, right? So here it's based on cleavage. The cell has to cleave itself into numerous cells, and during the process of cleavage, if any part gets destructed or if the organism loses any part, and if the organism fails to develop into a complete organism, then that is known as mosaic type of egg. Whereas regulative egg, if you damage any number of cells, the others compensate to the damaged one. So such type of eggs are found where one, if you damage a portion, then that portion is absent from the developed organism. Whereas regulative one, if you destruct any number, if you damage any number of cells, it develops into a complete one, because there it's not predetermined. Whereas in mosaic, it is each portion is predetermined or destined to become a part of the organism. Whereas regulative one, it compensates. And again, one more classification is there, and this is based on the presence of shell. Presence of shell, you can call it as cleidoic egg or a non-cleidoic egg. If there's a hard shell like of a chick, then that is known as cleidoic egg. Or if that hard shell is absent, then it is known as non-cleidoic egg. So this is the structure. When we say about egg, this is what comes to our mind, right? And this portion is what is known as ovum. Ovum with an egg membrane, and then this forms what is known as egg. And here you can find, this is the place which is the germinal portion, which is destined to become the embryo, and this lies on the animal pole, within the animal pole. So this region is animal pole and this becomes the vegetal pole. Here you have an air space which is filled with air, so that exchange of gases takes place between the egg as well as surrounding. This is a structure. You could find concentric rings of yolk. Yolk is there, plus an albumin is present, and this albumin, it's a thicker one. It's a thicker one that protects this yolk, and this keeps intact the nucleus. Nucleus is present in this region where you have the growing embryo, and growth is by means of this yolk. Yolk is again divided into two types, white yolk as well as yellow yolk. You could see the concentric rings. You have nucleus of Panter there, which is connected here to the latibra, to this latibral region. Then there are few structures like a chaliferous layer, and a chalaza is present which keeps the yolk intact. Membranes are present. These are membranes that cover and protect this egg, that helps to hold the nucleus in place.